Are there any more quizzes out? Yeah. Okay, I don't know how many of these the printer successfully made because it stopped kind of mid handout, so. It, no, we may not have enough for everybody, but if you don't, then please share. Because I couldn't stand there and wait for the printer to decide. Okay. We ended last time talking about these barcodes that are on the surfaces of everybody's cells. And there's two main classes of these. So these proteins, these HLA proteins, they fall into two classes. Class 1 proteins, class 2 proteins. These are encoded on a very large complex of genes that are clustered together on chromosome 6. This gene cluster is called the major histocompatibility complex. To illustrate the importance of these proteins, let's take an example. And it's a silly story, but hopefully this will help you visualize what these do. So let's say that Madison here is in an accident and she gets a burn and her arm is burned and she has to have skin debrided and she's just left bereft of skin. So let's say Madeline over here, being the wonderfully humanitarian person that she is, says, I'll give you a little skin graft so that you can have a piece of skin and it'll grow and it'll cover your whole arm. And our first names are almost the same, so they'll probably match, right? Well, there's more to it than that, obviously. So we take a little patch of skin from Madeline, we give it to Madison, and it sits on her arm and it seems to embed and everything seems to be great. But then there's a problem. After about a week, the edges of the graft start to retract. The graft itself then starts to discolor, and then the graft shrivels up and then it falls off. That is called graft versus host response. Graft versus host response. So the barcodes on the surfaces of Madeline's cells were viewed by the immune system of Madison's cells. And they said, these are not us. These are not self. Because they're not self, they're foreign. They're an invader. They need to be attacked. They need to be destroyed. But the skin graft protested, I'm here to cover your skin. To which the immune system said, so you're the wrong barcodes, so you need to be rejected. This is how your body distinguishes between what is you and what is not you. In other words, what is self and what is not self. So I think you can agree, major histocompatibility complex does not roll off the tongue very easily. So we're going to abbreviate that. We're going to call it MHC. There's two classes, MHC class 1 and MHC class 2. Class 1, class 2. The genes that encode these proteins are wildly polymorphic. Meaning they have tremendous genetic variation. So much so that is very unlikely for two of you in this room, in fact, two of you in the whole school, to have an array of MHC proteins on the surfaces of your cells that are the same. That's how polymorphic they are. The best way to get a match is to look to people in your family, because their genes come from the same source, that is, your parents. So, class 1 proteins are expressed on the surfaces of all nucleated cells. Class 2 MHCs are expressed on the surfaces of cells that present antigen. So dendritic cells will have them, macrophages will have them, B cells will have them, and other cells that present antigen will have them. None of the other cells will have them. So special cells have these. All nucleated cells have these. So will red blood cells have these? No, because they're not nucleated. Will platelets have these? No, they're not nucleated. But all other nucleated cells will have these. So will liver cells have these? Sure. Will skin cells have these? Sure. Will spleen cells have these? Sure. Will your lung cells have these? Will the cells in your big toe have these? Yes. All nucleated cells have these. All right. Is that part clear so far? Is there any part about that that you need that bears repeating? Your book at this point leaves these. I wish it didn't. I wish it would just tell you what they're for, but it leaves these. These are going to present antigen, and they present two types of antigens because of the way the cell treats them. We'll come back to that because of the way these slides are organized. Now, there are another group of molecules called cluster of differentiation proteins. And cluster differentiation is mercifully abbreviated CD. CD. CD proteins become especially important when we talk about T cells. So the three most important for our purposes when it comes to T cells are CD3, 4, and 8. All T cells have CD3. CD3 forms a complex with the T cell receptor. Without CD3, you do not have a fully assembled T cell receptor. T helper cells express CD4. CD4. Cytotoxic cells express CD8. T cells do not express, mature T cells do not express both of these molecules, they express one or the other. During their development, they'll express two of these. But then they'll downregulate one and upregulate the other while they're in the thymus.
Sure, those three then.
CD3, CD4, and CD8.
And which three, like what? You said
CD3 is complex to the T cell receptor. So all T cells have CD3. CD4 is only expressed on T helper cells. T helper cells. CD8 is only expressed on T cytotoxic cells. T cytotoxic cells are sometimes called killer T cells. Killer T cells. That term has kind of fallen out of use as the term killer becomes less PC. But it nicely describes what they do. Because if you're expressing viral antigens, that's exactly what the killer T cells do. They come up and they see to it that you off yourself. Now, what do B cells have on their surfaces for binding antigens? They have antibodies. Wait a minute, I thought antibodies were secreted. They are. But there are also antibodies that are on the surface. And the surface-bound antibodies act as the receptors to bind antigens. So T cells have the so-called T cell receptor. B cells have membrane-embedded antibodies. Both of these are capable of binding antigen. When they bind antigen, the B or T cell becomes activated. And the activated B or T cell divides. These antigen receptors are quite unique. And they're quite unique because of the way that they are made in your body. When B and T cells are activated and they divide, they make a clone of themselves. A clone of themselves. So only the B cell that has an antibody on its surface that recognizes an antigen is the one that will divide and make lots of copies of itself, make clones of itself. Same thing with T cells, only the T cells, whose T cell receptor happens to recognize that antigen, will be the particular one out of the whole population of T cells in your body that will end up dividing. As these cells divide and expand, some of those cells, as these cells start to mature, will be sectioned off and will become memory cells. When they become memory cells, they essentially go into a quiescent state. Quiescent means they take a nap. They take a nap until they're needed again. How do you wake them up? So think about how you get woken up in the morning. You either get woken up by the alarm that you set on a device like this one. Although yours, I'm sure, is a lot nicer than this one. Or maybe it's your roommate poking you saying, it's time for class. Get up, get up, get up, get up. Or if you are like my brother, while I was growing up, my twin brother, in fact, Tom, if he woke up before I did, would actually get on top of my bed, put his feet on either side of my pillow, and start to do this. And that eventually woke me up, because my brother and I were both pretty sound sleepers. You guys know I was born and raised in California. California has these things called earthquakes. When I was in graduate school at University of California, Irvine, which is in Southern California, we had the big Northridge earthquake. I slept right through it. Don't remember a bit of it, only when I got up and heard on the radio about the earthquake that had happened just an hour before. Oh, we had an earthquake. Oh, okay, whatever. So how do you wake these cells up? You bind something to their antigen receptor. When you bind something to their antigen receptor, an alarm clock goes off inside the cell. And the cells wake up and they start to divide. So that's their wake-up call, antigen. So for example, you all received your COVID-19 vaccines at some point in time. Those antigens selected a group of B and T cells, they expanded. Some of those got sectioned off for memory. And then when you had a second shot, That second shot essentially caused another expansion, and you had other cells essentially that were put aside as memory cells. And now you've got memory cells, so if you get exposed to it again, those cells will expand and your infection will either be asymptomatic or it'll be pretty minor, because now your immune system remembers, it's primed. So it's just like seeing that patient who's got the same illness. You know what to do now. You're primed because you went through it once before. That's kind of the way your immune system works. The adaptive response shows memory. Jenna?
So if the B or T cells weren't able to carry on memory, would that kind of lead to the immune compromise?
If they're incapable of making memory cells, sure. I can't really think of an immunodeficiency that just affects memory. It usually affects something more basal. But there are things that will prevent the immune response from being completed. Therefore, it prevents certain cells being set aside as memory cells. All right, so your T cells come in three main flavors. There's actually more than these, but these are the three main ones for our purposes. T helper cells, they express CD8. Regulatory T cells, helper cells express CD4. Cytotoxic T cells that express CD8. And then so-called regulatory T cells. And regulatory T cells express CD4 at first, and then they downregulate it. Then they express something called CD25. These cells essentially turn the immune response down. This is why the immune response, when the amount of foreign substances, the amount of pathogens go away, doesn't keep going. It tailors off, and that's due to largely the effects of these regulatory cells. T cells don't make antibodies. So helper cells will activate other cells. So they activate B cells, they activate cytotoxic cells, and they activate macrophages. Cytotoxic cells sniff for foreign antigens on the surfaces of cells. They find a cell with foreign antigens, they bind to it, they make poisons, and they destroy it. That's why they're cytotoxic. Regulatory T cells secrete cytokines that downregulate the immune response. So don't get it in your head that all cytokines activate the immune response. No, cytokines regulate the immune response. Some upregulate it, some downregulate it, and some turn it this way or that way or whatever. They regulate the immune response. B cells, when they bind antigen and are activated, they will divide and they'll start to secrete. When they're activated, that antibody that's on their surface, they now make a version of it that's secreted rather than membrane bound. And those antibody secreting B cells are called plasma cells. If any of you work in oncology, you'll hear about things like plasma cell cytomas. Plasma cell cytomas are tumors of plasma cells. Multiple myelomas, those are tumors of plasma cells. And the antibody that they bind to the antigen serves to do several things to the antigen. First of all, it clumps the antigen by clumping it together. It's more easily recognized and it's more easily destroyed. So it clumps it, and that helps cells recognize it and destroy it. 2, they opsonize it. Opsonize it. What's opsonization mean?
It's when it creates the handles on it, so that it keeps using.
You put a handle on it, so now cells can phagocytize it and destroy it much more easily. Put a handle on it. Now it can phagocytize it and destroy it much more easily. That's what opsonization does. So phagosomes have on their surfaces receptors for antibodies. We call those receptors FC receptors. When the antibody is bound to the antigen, they can bind these receptors and it'll help the phagosome take it up. Third thing they do is they neutralize the antigen. If your antigen's a toxin, you bind it with antibodies, it's usually not toxic anymore. If it's an enzyme and you bind it, the enzyme usually doesn't work anymore. So it clumps it, it will opsonize it, it will neutralize it. And it in general, essentially think of an antibody as a guided missile. A very specific guided missile for your antigen. That might be a good way to think about it. So B cells mature in the, T cells mature in the, and both cell types then migrate to the, right. Your lymph, well, they go to the blood usually. They'll go to the bloodstream usually, and then they'll usually go to the lymph, but they end up usually in lymphoid tissues, like the lymph nodes, like the spleen. So, bone of the bone marrow, T cells mature in the thymus, they go to the lymph nodes and to the spleen after being released into the bloodstream. And then going from the bloodstream to the lymph nodes and the spleen. Here are some differences between B cells and T cells. So the first two we've already talked about. In blood, you don't usually see very many circulating B cells. They're usually in the lymph system. They're usually in lymph nodes, in fact. T cells you'll see in higher numbers, usually circulating in the blood. The receptors for antigen in B cells are your surface immunoglobulins. T cell receptor, the T cell receptor is not secreted. It's not secreted. If you have a secreted T cell receptor, you've got an abnormal T cell receptor. It is not secreted, it's a cell surface molecule exclusively. In lymph nodes, B cells are found in high numbers in the cortex of the lymph node. T cells will be found interior to that. So on that, the periphery of the cortex shows the B cells. T cells will be found inside the cortex, so-called paracortical sites. Do they require antigen to be presented with major histocompatibility complex proteins? T cells do. B cells, not really. They need T cell help to be activated, but the antigen, they don't need it presented to them via MHCs. They can have it presented to them, but they don't need it. when you activate a B cell, you make a plasma cell and memory cells that secrete antibodies. T cells, you get your activated types of T cells and then memory T cells as well. Memory T cells can be really, really important for certain types of diseases. So for example, you guys were immunized hepatitis B years ago, over a decade ago. If we were to take blood from you today, none of you, I'll bet you, would have any antibodies to hepatitis B. Yet all of you are immune to hepatitis B because you have memory T cells. General functions. B cells make antibodies to the antigens that inactivate, neutralize, and target them. T cells, it's to either activate or suppress other cells, to kill abnormal cells in the case of T cell toxic cells, and to mediate hypersensitivity under certain conditions, and to make a lot of cool cytokines that regulate the immune response. Okay, this is a recap of what we said about T cells. Is there anything on here you need me to elaborate on that I haven't already elaborated? No, then I will move on to the next bit. So here's the T cell receptor. The T cell receptor does not sit in the membrane in isolation. It instead is a member of a complex. So the T cell receptor has two polypeptides, an alpha and a beta polypeptide chain. And it forms this dual molecule, and it has a cleft right at the top, and the cleft is the antigen binding site. Now, if you please take the handout from today. How do you get T cell receptors essentially that can recognize so many different types of antigens? The answer has to do with the way the genes are actually on your chromosomes rearranged to make this specific type of T cell receptor. So, turn to, let's see where. Oh, here it is, here it is. Okay, page it looks like this. It's page 1, 2. It'd be page, I guess, 4. No, 5. So at the top of this page, you have essentially the alpha chain locus in your body. So you have this region, it's called the variable region. There's 70 to 80 different types of variable regions in the alpha chain locus. 70 to 80 different types of variable regions. Then you have these so-called joining regions, and there's 61 of those. Then you have a so-called constant region, you only have one of those. Now the constant region is the piece that sticks in the membrane, like that. So that piece is not going to encounter antigen, is it? These pieces up here are the pieces that will encounter antigen. And they're encoded by one of these 70 to 80 different types of variable chains and one of these 61 different joining chains. So what your body does is it selects one, during T cell development, it takes one of those variable chains, and it picks one of those joining chains, and it takes that particular variable chain and sticks it right next to one of those variable chains. All the other DNA in between, it's looped out and cut out and thrown away. And the way your body essentially does it is shown right here. You essentially pick one of these, one of those, you stick them together, the rest of it, you loop it out, cut it out, and then that loop gets degraded. And so that means essentially that now this cell will make a specific type of alpha chain that has one of these V segments and one of these J segments. With that particular constant region. Then it'll do the same for the beta chain. With the beta chain, you have 52 different variable segments. And then you have a diversity segment next to these different joining segments. You have 6 of those with a particular type of constant region. And then a second diversity chain with 7 different of those joining chains. You're going to pick one of those variable chains and join it to this diversity chain and one of these joining chains. That'll be matched to one of these particular types of constant regions. Then you're going to pair this chain with that chain to make a mature T cell receptor. Now, how many different types of T cell receptors do you think you could make with this many different segments? It would be, let's say 80 times 61 times 52 times 13. Because there's 13 different types of J chains there. Times 2, because there's 2 different types of diversity chains. I don't have my calculator up to do that, but what you get is a really big number. And every T cell essentially will have one of those very big number of different types of T cell receptors on its surface. And one of those T cell receptors will usually be able to bind the antigen that is presented to it. And only the one whose T cell receptor recognizes that antigen is the one that will start get activated and start to divide. So this type of rearrangement, this rearrangement is due to 2 proteins that you express in your T cells and B cells. These are called recombinase. So the recombinase, there's a recombinase 1 and a recombinase 2. And they're called RAG1 and RAG2. And if you don't make RAG1 or RAG2, you cannot rearrange your T cell receptor genes, and you cannot rearrange your immunoglobulin genes. You don't have any diversity of antibodies. You don't have any diversity of T cell receptors. Well, no, it goes beyond that. You don't make a T cell receptor, and you don't make any antibodies. And you have something called severe combined immune deficiency, SCID. And you have to live your life in a bubble, because even the common cold can kill you, because you have no adaptive immunity. And that's what deficiencies in RAG1 and RAG2 will do. All right, so this is shown essentially on this page down here. So you rearrange the DNA while the T cell is developing. You make a specific type of alpha chain. You rearrange the DNA that encodes the beta chain and make a specific type of beta chain. You pair those together, and that gives you this very specific type of T cell receptor That is then paired with CD3. When you pair it with CD3, you can bind antigen. Now the cell knows that you're expressing a proper T cell receptor. So this essentially table here on the facing page shows you how many different types of T cell receptors you can make. So the total diversity of T cell receptors that you can make is 1 times 10 to the 18th. That's even bigger. That's an even bigger number than our national debt. You can believe that. I didn't get any last year. I didn't get last year. That's a big number. How many antigens will you encounter in your lifetime? Will it be that many? No. This is one of the reasons why these people who say kids get too many vaccines too early, and there's a group called Too Many Too Early. How many antigens do kids experience when they come out of the womb? A heck of a lot more than they'll ever get in a shot. You guys ever seen what kids put in their mouths? If you haven't, wait till you have your own. Because the mouth essentially is the way they experience their world for about the first, well, wait till Rachel, the first 6 years of her life, for about the first 2 years of their lives. They put dirt in their mouths, they put their hands in their mouths, they put their toys in their mouths. You give them a book to read, it usually goes in their mouth and what have you. So they experience far more antigens on a day-to-day basis than they ever experience from the vaccines that they get. So that victim, too many, too late, makes no immunological sense whatsoever. None. Zero, zip, nil, nada is a statement born of stark ignorance. As well meaning as these parents might be, they are ignorant. So if someone tells you that during your practice, please explain to them why that should not be a concern. It really shouldn't. Given the flood of antigens that kids are exposed to every hour of every day, what they get in a shot is small potatoes. So you guys, if you guys heard the news about Aaron Rodgers being diagnosed with COVID-19, and he said in an interview, I was immunized, Cassie, you're laughing. Did you see that interview with Aaron Rodgers?
Me?
Yeah, you're laughing at it. I thought you saw it.
Sorry.
Did you see the interview with him?
I heard about it and I think possibly about Carrie Underwood. I don't know. I saw it on Facebook. Yeah.
Carrie Underwood?
Yeah. Anyways, yeah, Carrie.
Right, no, I'm not sure Carrie Underwood had anything to do with it. So Roger said in an interview, he was asked by a reporter, have you had the COVID-19 vaccine? He said, yes, I've been immunized. In his reading, Rogers read about the vaccine and he thought he saw in the list of components in the vaccine, a component to which he was pretty sure he was allergic. So he didn't want to take the vaccine because he was afraid of an adverse reaction to the vaccine. Why he didn't take the Johnson & Johnson vaccine, I don't know, but that's another story. So he went to a homeopath who did some sort of, let's be honest, voodoo to immunize him. And he said, yes, I've been immunized. Then he's diagnosed with COVID-19, and it turns out he kind of shaded the truth in that interview. So now it turns out the team knew about this, and so did the NFL. And they did nothing, but it's somehow his fault. That's the part I can't figure out. So he was fined $14,000. Now, $14,000 to me is a lot of money. How much is it to a guy who makes $6 million a year? Peanuts. That's exactly what shots are to a kid's immune system, given the amount of antigens they're exposed to day in and day out. It's like define the NFL via generic properties. There's my pop culture reference for the day. This always goes off better for the guys because they're usually up on football and stuff like that. Anyway. All right, so that's essentially how you get a T cell that has that kind of diversity because of this literal rearrangement of the genome of these cells while they are developing. It's a really remarkable sort of event. All right, let's now go to the B cells. Very good. So let's take the first page here. So a little bit about antibodies. And you have another handout with some pictures of antibodies on them. And I just want to show you, so there's this piece of pizza. So antibodies, they're kind of Y-shaped. The end of the two bits on the Y, the tines, if you will, on the fork, so to speak. This is the piece that binds the antigen. That's the piece that binds the antigen. And those pieces are variable from antibody to antibody. This piece on the back, the common part of the Y, that is called the F sub C portion. The F sub C portion or the FC portion. It's called FC because if you clip the antibody in half, that piece will crystallize. So it's the fragment that crystallizes. That's why it's called F sub C. That's the piece that interacts with receptors on the surfaces of cells. That part doesn't bind antibody, doesn't bind antigen. This piece on the ends of the Y, they bind the antigen. Now, if you look at these three pieces of the antibody, they're all encoded by different parts of the antibody gene. So notice that the antibody is composed of two polypeptides, a heavy chain and a light chain. There's two light chains and two heavy chains, and they're linked together by disulfide bonds to make this Y-shaped structure. So let's talk about the light chain first. The light chain is encoded by a variable region, a joining region, and a constant region. A V, a J, and a C. Variable region, joining region, constant region. What about the heavy chain? It's encoded by a variable chain, variable region, a diversity region, a joining region, and then a set of three different constant regions. The variable region, a diversity region, a joining region, and then a series of different constant regions. Now, how many of these different types of variable regions, joining regions, diversity regions do we have? Look at the table, please, we'll know that. There's two different types of light chains, kappa and lambda. Kappa and lambda. You have 40 different variable kappa chains. You have 30 different variable lambda chains. You have 5 different joining chains for the kappa, 4 different joining chains for the lambda. In the heavy chain, you have 40 different variable chains and 25 different diversity chains and 6 different joining chains. That can generate a lot of diversity. A lot of diversity. All right, turn the page, please. And here you have the way these essentially are arranged on the chromosome. So for the heavy chain, you'll pick one of those variable chains, just one. You'll pick one of those diversity chains, just one. And you'll pick one of those joining chains, just one. You'll take one of those variable chains put it next to one of the diversity chains. You'll take one of those diversity chains, put it next to one of those joining chains, and those will be paired with the constant region. And that's essentially the particular antibody that you'll make. That can generate a lot of diversity. And then you'll wash, rinse, repeat for the light chains. You'll pick one of those variable chains, put it next to one of those joining chains for the kappa chain, one of those variable regions, put it next to one of those joining chains, and to either one of those variable constant chains, so on and so forth, for the lambda light chain. And the next page essentially shows you how this does this. Essentially, the recombinases, they just pick one. Turns out there are little cues in the space between the V and the J and the D. So these little sequences here, those are the kind of handles that RAG uses. So it grabs these and it joins them together and it clips the intervening piece out, and then it joins those two together. And when it joins those two together, that joining those two together is an error-prone phenomenon. Because it's error-prone, you get even more diversity in the way you're able to generate these antibodies. Now, this table here that we use for our T cells, this will calculate essentially the number of different types of antibodies that you make. And given all these different types of sequences, you make 5 times 10 to the 13th. That's 50 trillion different types of antibodies, but that doesn't include these mistakes that you make while you join it together. So it's actually greater than 50 trillion types of antibodies. That's a lot of different types of antibodies, isn't it? Your excitement overwhelms it. So that essentially is what's going to be on the surface of your B cell. Is this particular antibody that was made essentially from that series of events. That's how you get the diversity of T cell receptors. That's how you get the diversity of antibodies. And this happens in the B cell, it happens in the bone marrow. And if you don't get a properly rearranged heavy chain and a properly rearranged light chain so that they can make these chains that can be linked together and be expressed on the surface of the cell dies. It does not go on to become a B cell. Only mature B cells that make functional antibodies are released from the bone. In the thymus, only T cells that have a properly rearranged set of alpha and beta T cell receptor genes that make a proper T cell receptor that is coupled with CD3, only those cells will then go on for screening. And if they don't react with self-antigens, they'll be released from the thymus. You don't properly rearrange those genes, the T cell gives a signal to that cell to off itself, and the cell will die. It's very stringent. So only the strong survive. Well, only the properly rearranged genes survive when it comes to these lymphocytes. I don't pretend this is simple. I know some of you are giving me these forlorn looks saying, what have I gotten myself into with this nursing thing? This is among some of the hardest stuff that you will study as nurses. Easily. But the immune system, it's because essentially you've got this variability. And what kind of antibody will you make? Well, it's a bit of a dice throw, isn't it? But essentially through this seemingly chance event, you get this incredible diversity that can handle all kinds of things that are thrown at you. From the antigens you experienced when you first came out of the womb to the antigens you'll experience on your deathbed when you take your last breath. Your immune system is constantly finding ways to deal with new pathogens. All right, let's take our break, 5 minutes, and then we'll come back and talk about how these are used in your body to fight disease. Yeah.
So when you were talking about the Aaron Rodgers and his homeopathic whatever, I didn't take this quiz yet. That's Cassie's. She's not here. You mentioned that he should have just taken the Johnson & Johnson one.
Right, because the tronilin in the messenger RNA shots, he was pretty convinced he was allergic to that.
Oh, okay. My dad is allergic to contrast dye and stuff, and his doctor told him not to get the vaccine yet because they don't want him to have a reaction to it. Kind of like,
He might, of some of the pegylated lipids, he might
yeah.
respond to that. Now they can patch test him
Yeah.
first, or he can get vaccinated in a hospital.
Yeah.
And if he has an anaphylactic shock, hospitals are very good at treating that.
Yeah, he had it. My dad has cancer, and he had some sort of scan a couple years ago, and they put contrast dye in him, and he almost died.
Yeah.
It was pretty bad.
Yeah.
Anyways, that's how it is.
That's why you're supposed to patch test people first.
So then you know. Another thing was you posted that article about infertility and pregnancy and the COVID vaccines. My cousin's pregnant right now, and she actually just texted my mom. My mom's a nurse, and was asking her if she should get the vaccine. Like, she's not sure. Does she have any experience with people getting it? And then I read that article, so I sent that article over to her, and I was like, "Hey, my microbiology professor sent us this article. Pretty trustworthy sources, like this, that." She's like, "Thanks. Like, I think that's pushing me towards like getting the vaccine." So those articles that you post are very helpful. And I wasn't even expecting that. I was just like looking through Blackboard, and I was like, "Oh, that's very."
See, I think the CDC just needs to say, look, we have good preliminary data on this. It says it's safe during pregnancy because during the phase 3 trials, there were people who were pregnant.
Yeah.
They didn't know they were pregnant.
Yeah.
They were tracked and they went on to give birth to healthy babies. Their miscarriage rates weren't any higher than those who got the placebo.
Yeah.
Since then, it's been continued to be tracked with a cell phone app. And once you get vaccinated, you get a questionnaire. And if you're pregnant, they will say, can we continue to track you? From that, there's data that's come out. It's about 1,000 people who've been involved with that. And the data so far says in the group that got vaccinated and were pregnant, their miscarriage chances aren't any higher. In fact, they're a little bit lower than the usual miscarriage date.
Yeah.
Yet you have people on Twitter saying the vaccine causes miscarriages. And the way they get the number of 80 some odd % is they take the number of people who miscarried and they multiply it by people, divide it by people who had problems in general during pregnancy. There's a word for that.
Yeah.
Dishonest.
Yeah.
It's completely being dishonest with numbers.
Yeah.
And I've heard pastors in the pulpit repeat that number. And I've emailed people saying, that's a lie.
Yeah.
You need to not lie at the pulpit. You need to go to front of your congregation and tell them that you misspoke, that you used wrong numbers.
Yeah.
And they've been very, very brusque with me. And I'm just saying, I told you, you will answer before God for this misinformation you're giving to your congregation. I don't think being a pastor somehow exonerates you from telling falsehoods.
Yeah.
In fact, I think you have a bigger responsibility behind the pulpit to truth.
Yeah.
So I told you,
Yeah.
if you don't care about truth, then
Yeah.
Jesus who said, I am the truth. Come on.
Yeah. Yeah. Like I've seen that before. Like you and you. Yeah. Oh yeah. So funny. Dress. Well, last night I was like trying to, I was trying to fit it, and I was watching this last night. It's like a short slider, but I need it. Yeah, I need it different. I need it different. Yes, I need my white.
Okay, let's get back in the saddle here. When a B cell or a T cell encounters an antigen that fits its T cell receptor or its membrane-bound antibody, and it becomes active, and you get this expansion of that specific cell, that is called clonal selection. Clonal selection. Clonal selection essentially guarantees that only those specific cells that specifically respond to this particular antigen are the ones that are activated and that expand and that divide that you get that you get more of. That's clonal selection. If those clones get destroyed during development, so some sort of random genetic issue prevents you from making certain types of lymphocytes because of rearrangements in the genes that encode the T cell receptor or immunoglobulins, that can prevent you from making certain types of T cells and B cells. That is called clonal deletion, and that can have some pretty remarkable consequences, immunologically speaking. Clonal deletion also occurs during the development of B cells and T cells. Even mature B cells and T cells that make a proper T cell receptor, that make proper immunoglobulins, if they react with self-antigens, they will be destroyed. And that is another form of clonal deletion that actually prevents you from making T cells and B cells that react with self-tissue. So for each of the properties listed, determine if it describes a T cell or a B cell, or mainly, or maybe both. So it matures, not born, matures in the bone marrow. B cell or T cell? B cell, B cell. T cells are born in the bone marrow, but they mature in the thymus, thymus. High numbers of cells circulate in the blood. T cells, T cells. B cells tend to be introverts. They're shy. They hide in the lymph nodes, whereas T cells are the extroverts out in the blood, shaking hands with everybody. And if you express bad antigens, they will probably kill you. But they're the extroverts. So B cells are the introverts, T cells are the extroverts. Is any of you cold? It is a little cool in here, isn't it? You keep hunkering down, trying to make yourself small. They require antigen to be presented with an MHC molecule, B cells or T cells. T cells usually, B cells sometimes they can have antigen presented by an MHC molecule, but they don't require it. So require is the money word here. They produce antibodies, B cells or T cells. B cells. Antibodies, B cells, antibodies. They're produced in the bone marrow, they mature in the bone marrow, B cells. Cells are activated to help other immune cells suppress or kill abnormal cells, etc. B cells or T cells. Definitely T cells. Definitely T cells. Now, let's talk about presentation of antigens. When your body experiences a foreign invader, those antigens that it'll experience are usually parts of the whole invader. So these are bits of the cell wall, things that the cells may secrete, complex protein, glycoproteins, complex carbohydrates, so on and so forth. Some polymers will be more immunogenic than others. It depends, but It's usually some little piece from the cell that it's going to respond to. Certain things are going to be more immunogenic than others. Proteins are usually pretty immunogenic. Glycoproteins and lipoproteins will be less immunogenic, but still, they're pretty good antigens. Polysaccharides are usually pretty miserable antigens. Nucleoproteins, those are proteins complex to DNA. Pure DNA is a terrible antigen, but DNA complex to protein, now you're talking. That's a pretty good antigen. Lipopolysaccharides, it depends. Some lipopolysaccharides, terrible. Others, pretty darn good. We're going to look at an organism that causes undulating fever called brucella. Those of you who grew up on farms have probably heard of brucella. Your cattle have to be immunized against brucella. Brucella has a lipopolysaccharide that's a lousy antigen, terrible. Your immune system hardly responds to it. Other types of gram-negative cells with their polysaccharides, E. coli, salmonella, their lipopolysaccharide is an excellent antigen. So some are good immunogens if they have lots of variety in their structure. The more repetitive you are, the less effective you are as an antigen. And you have to be usually a certain size. The bigger you are, the better an antigen you are. And we'll see why in just a bit. So here are your lousy antigens. If you repeat a lot, not very good. Sugars, especially short ones, terrible. Proteins, quite good. Pollen, very good. Foreign animal cells, things like that, whole cells, very good, and so on and so forth. Now, if your molecule is too small to elicit an immune response, then you stick it on something that is a good immunogen, and that carrier now will cause the immune response to be made against it and the thing is carrying. That's called haptenization, and the hapten is the bigger thing that's carrying the small bit. And this is used essentially in some vaccines. So the pneumococcal vaccine that you received as kids was haptenized. The capsular carbohydrates, they're sugars, they're repetitive, so immunogenicity, essentially, so they're attached to a carrier, then injected. That's called a conjugated vaccine. And those are now very good immunogens. Alloantigens simply refer, allo means other. Those are antigens from sources other than you. That's an alloantigen. Alloantigens are responsible for rejection during organ grafts. They might also cause incompatibilities in blood transfusions. That's why you always need to cross-match your blood before you give it, even if you're in a hurry. Superantigens elicit over-the-top immune responses because of the way cells interact with them. We'll take a look at those. I doubt we'll get to it today. We'll probably do it on Monday. And then allergens evoke allergic reactions. And not only in chapter 14, but we're probably going to have to skip chapter 14 out of the interest of time. Okay, let's look at antigen presentation. So your three main antigen presenting cells, APCs, are dendritic cells, macrophages, and B cells. Dendritic cells would be number one in importance. Macrophages would be a close second, and coming in a solid third would be B cells. That's not to diminish B cells, but dendritic cells are definitely the most important. So here's how they do it. So the antigen is going to essentially be bound to the surface of the antigen presenting cell. Now, typically, these cells will phagocytize the microbe or whatever, and it will fuse it with a lysosome, and the lysosome will break it into lots and lots of pieces. So this piece then is going to go from the lysosome, and it's going to be kept in this little vesicle called an endosome. The class 2 major histocompatibility complex protein is now going to be moved from the from the Golgi apparatus. It's going to be moved into that endosome. And that endosome then is going to bind that antigen. And then that MHC2 protein will be trafficked to the surface of the cell. And now your MH2 protein is going to essentially present this on the surface of the cell. So that means that MH2 class 2 presented antigens come from things that were at one time in the lysosome. They're the product of degradation of microbes. They're the product of degradation of microbes. They're things from outside the cell, they came into the cell, they got destroyed, they got chopped up into little bitty pieces, and then they got bound by these class 2 molecules and then sent to the surface. And now, essentially, this is how the T cell receptor will perceive the antigen. It'll perceive the antigen served on a platter of the class 2 MHC. That's what we mean by antigen presentation. The T cell is just not going to react. It's not going to go off half-cocked and respond to any old antigen. It needs a license to respond to that antigen. What's the license? The license is that it needs to, number one, have that antigen given to it by a class 2 molecule. And notice that that class 2 molecule also interacts with the CD4 protein on the surface of the T cell. So you need that contact between the T cell receptor and the antigen bound by the MHC2. And then the CD4 molecule also needs to be in contact with the MHC class 2. You must have both of those contacts. When you have both those contacts, you get a signal that goes into the T cell, and the T cell becomes activated and now starts to undergo clonal expansion. That's how you activate a T helper cell. The antigen has to be presented to it in the context, as we say, in the context. It has to be presented to it while it's bound to class 2 MHC protein. And only antigen presenting cells express class 2 MHCs. So therefore, it can only respond to antigen presenting cells. You can give it all the soluble antigen you want, it's not going to respond to it. It has to be presented to it by these antigen presenting cells with it bound to the class 2 MHC. The cutting up of the antigen into smaller and smaller bits that are bound to the class 2 MHC, that's called antigen processing. Antigen processing. And then the binding of it and the trafficking of it to the surface, that's called antigen presentation. Antigen presentation. And these are just some of the examples of things that might be bound to the MHC class 2 protein. So which of the following is not an antigen presenting cell? Is a macrophage an APC? Yeah. Is a dendritic cell an APC? Yes, that's one of probably the most important APC. Is a T helper cell an APC? No. A B cell is an APC. T cells, they have antigen presented to them. They are not APCs. So the answer there is C. All right, so the T cell, depending upon the type of antigen that is presented to it, will respond in one of several ways. It's going to make cytokines. Now, one of the first cytokines it makes is interleukin 2. Interleukin 2 is going to act as a T cell activation signal. By making interleukin 2, it causes itself to divide and to expand. So here are some of the different subtypes of T cells. Even the T helper cells, there's actually two types of T helper cells, a T helper 1 and a T helper 2. Here's our buddy here, the cytotoxic T cell. They express CD8 on their surface. The T regulatory cell, they express CD4, but they also express CD25. A so-called T follicular helper cell has CD4, and then this other thing called the ligand for CD40. And then this other cell called a T helper 17 cell that also expresses CD4 as well. And then these dudes down here that respond to lipid antigens and gamma delta T cells. We'll talk about those later. These are a minority group of T cells. So each of these is going to respond a little bit differently. So let's talk about these folks here first. All right. So the helpers, they secrete interleukin 2. That is going to auto-stimulate the T cell. The other thing they will express is interferon gamma. Interferon gamma is a signaler for macrophages. It gets macrophages all hopped up and ready to go. Other cytokines that they might make are interleukins 4, 5, and 6. Interleukins 4, 5, and 6 will stimulate B cells. So the T cell will stimulate itself. Interleukin 2 will also stimulate T cytotoxic cells. It makes interferon gamma, which stimulates macrophages. And it makes interleukins 4, 5, and 6 that activate B cells. Cytotoxic T cells will be able to bind to proteins that are expressed on the surface of the cell that are the result of viral infections. Those viral infections will bind to the T cell receptor, they'll activate the cell, and the cell will now make these poisons. And those poisons, those cytotoxins, those lymphotoxins, will kill the virally infected cell. Cytotoxic T cells will also destroy cancer cells because cancer cells will make weirdo proteins on their cell surfaces sometimes. This is the basis behind a new therapy for certain types of cancers called immunotherapy. Is to get the cytotoxic T cells to wake up and recognize the cancer. Once they recognize the cancer, they'll destroy it. And so this is a therapy that's proving remarkably effective against certain types of tumors that are resistant to lots of different chemotherapeutic agents. Now, cytotoxic T cells, however, can be activated by antigen presentation of a different type. If a cell is infected by viruses, my red marker. Somebody took my red marker. Papa Bear is not happy. Let's say we have a virally infected cell. Those viral proteins very often are going to be expressed in the endoplasmic reticulum. Guess what else is expressed in the endoplasmic reticulum? Class 1 major histocompatibility protein. In the ER, these viral proteins will end up being bound to class 1 proteins. And then those class 1 proteins get trafficked to the cell surface where essentially they will have this viral protein presented out there like that. So this cell essentially has this viral protein, and this T cell receptor on the surface of the cytotoxic T cell can recognize that viral protein in the context of this MHC class 1, and CD8 is able to also interact and form a contact with that class 1 protein. So these class 1 proteins are proteins that actually come from inside the cell. Whereas class 2s were from things that came outside the cell and then ended up in a lysosome, were chopped up, and then those were bound to a class 2 that were delivered to the lysosome and then sent to the surface. These actually come from things that are made inside the cell. Made inside the cell. That's why class 1 MHCs will typically present viral antigens. Viral antigens, because the virus, remember, co-opts the cell machinery to make its own stuff. That then gets sent to the surface, and that essentially will activate the cytotoxic T cell. That will then make these molecules that will kill this cell. And this cell then will die. By killing the virally infected cell, you prevent it from releasing mature virus particles. That cell then can be replaced by other cell populations and so on and so forth. And these cells, remember, are activated by interleukin 2 that's made by T helper cells. T cytotoxic cells can make their own interleukin-2, they just don't make very much of it. They need T helper cells essentially to come in and give them the help. Gamma delta T cells have a completely different type of T cell receptor. Their range of variability of T cell receptor is low. These predominantly will bind to lipid soluble antigens. They respond quickly, they do produce memory, and they react to certain types of phospholipids. They'll react to tumor cells because tumor cells will sometimes spin off these oddball phospholipids and things like that. So they are kind of a little bit between innate and adaptive immune response in some ways. Another group of cells that are part of the immune response are the so-called natural killer cells. Natural killer cells. Natural killer cells do not have an antigen receptor on their surface. They do, however, look at MHC class I proteins. And if the MHC class I protein is presenting an antigen that is weird, the natural killer cell will kill the cell. What kind of cells make weird antigens that get presented on MHC-1s? Virally infected cells and cancer cells. So these cells are some of the first cells that attack and kill cancer cells. And they respond to a so-called interleukin-12. Interleukin-12, they also respond to interferons. There's also a group of T cells that are called natural killer T cells. They have T cell receptors for antigen, and they act a lot like natural killer cells. So cytotoxic T cells are programmed to kill all the following except when they kill cancer cells, when they kill virally infected cells, when they kill foreign cells grafted in the body, when they kill bacteria. No. All right, now we get into antibodies. Very good. We'll finish this. On Monday, we'll probably skip 14 and we'll get straight into the different types of infections.
That's what I'm saying.